All right, here I have a picture of our gene that it consists of a lac operon here. We have our promoter, our terminator, and our regions in between that are considered the Z, Y, and A regions that will code for a particular protein. Well, the thing is, the RNA polymerase, which originally will stick down here, all right, your RNA polymerase sticks here, moves across this region until it gets down here, and of course it's going to fall off down here. But as it's moving through here, before it falls off, and before the RNA falls off as well, it's been making this long RNA that has the Z, Y, and A on it. And what we need to discuss is what happens. Well, if you remember, I said that in prokaryotic genes, now this was in class, prokaryotic genes, the ribosome finds the RNA. The RNA doesn't go out and find the, I should say, the mRNA doesn't go out and find the ribosome. That's what happens in our cells, in eukaryotes. All right, but instead, the ribosome, abbreviate that there, finds the RNA. All right, and so there are certain regions on here. There's a certain sequence, and I'll give you that sequence. Every so often along here will occur, just kind of like the Tatabox, only this is an RNA. And this sequence is called the Shine-Delgarno sequence. Shine-Delgarno. Or we abbreviate it SD. So these are all SD sequences right here. So SD, etc. All right, so all of those are Shine-Delgarno sequences. And what they do is the SDs attract the ribosome. And so a ribosome will attach here and start making a protein. But unlike a eukaryotic gene, where you only generally have one ribosome attaching at a time, or they'll be in sequence and they all attach at the beginning, in bacteria, every one of these, as they're made, all right, can attach a ribosome and make a protein. So if it's attaching here and moving in this direction, then it's going to be short. This one's going to be longer. The one that attaches here will be even longer than that. All right. Now, there will be places on here, stop codons, that will make these fall off. So between Z and A, maybe you get these. All right. But between Y and A, I mean between Z and Y, you get these three. But in Y, you'll probably attach and it'll start anew. I'm sorry, I made these. If this one has attached here and moved to there, it's going to be long. Realize they run over each other. So the first one, but here it's probably going to fall off. So right here, there's probably a stop codon. And so it probably has to start over. But if it starts over from there, it'll be short. It'll be a little longer. And this one will be a little longer. All right, and of course these are going to coil up into proteins, and then here you have one that's going to attach and start making the A protein. So all of these will eventually coil up into like Z's. These will coil up into Y's. These will coil up into A's. Now this is one of the traits of a polycistronic protein. Now if the stop was not there, it will keep on going and just make one really long one. And in some operons that's what happens, and you make this really long protein that coils up into this giant protein, and then other proteins come along and chop it up. Now that's an alternative system. All right, these other proteins will come along and cut out a piece and do stuff like that and make separate proteins. But that's not actually what the lac operon does. The lac operon works this way, and it waits for a stop, and then it breaks apart, and and you'll actually make Z's, Y's, and A's. Now we'll talk a little bit later about some things that are in between the Z and Y and the A, but that's going to be like the last video. And I don't know if it's the least important. It might be one of the most interesting things about how this can be regulated even more. But generally, you have Z, Y, and A, and you'll have multiple ribosomes attached all along here wherever there's a Shine-Delgarno sequence. And one of the things we'll be doing in this class is looking for these Shine-Delgarno sequences so we can do what we call annotate this gene. So just letting this occur, all right, I wanted to make sure you understood what was going on here and how different this is from what you learn about eukaryotes. So remember that in prokaryotes, the ribosome finds the RNA and it attaches. And where does it attach? It attaches at this thing called the Shine-Delgarno sequence. I'll give you that sequence later on. It's not that important at this point. It is when you do an assignment, and I'll give it to you in the assignment. And what you'll of course be doing, you'll be searching for that particular sequence all along your RNA. All right.